Students, one more application, biotechnological application in medicine. One more biotechnological application in a medicine, that is gene therapy. Gene therapy. That gene therapy technique developed by Anderson develops the technique of gene therapy in England. The gene therapy means the technique replacement of defective genes by normal functional genes to correct genetic diseases. That is called gene therapy. It is the technique in which replacement of defective genes. It is the technique in which replacement of defective genes by normal functional genes to correct genetic diseases. That technique that is called as gene therapy, commonly called gene therapy. This kind of gene therapy first given in 1990 to 4-year-old girl. This kind of therapy first given in 1990 to 4-year-old girl suffering from ADA deficiency. That ADA deficiency, due to the effect of ADA deficiency, the patient causes one kind of disorder, one kind of disease that is called as SCID disease. SCID means severe combined immunodeficiency syndrome. It is very dangerous disorder. What is that? ADA deficiency causes one of the disorder, that is SCID, severe combined immunodeficiency syndrome. Usually the ADA means adenosine deaminase. Adenosine deaminase is the enzyme. Adenosine deaminase is the enzyme. That enzyme is required for normal functioning of immune system, for the process of immune response, for the immune system, for the body defenses. What is that? This kind of enzyme is very necessary. This kind of enzyme activates lymphocytes are the type of WBCs. If the lymphocytes which are activated by adenosine deaminase enzyme, the lymphocytes which are activated, the person develops immune system. For the maintenance of immune system of the body, what is that? ADA enzyme. It means adenosine deaminase is very responsible. ADA is responsible for normal functioning of the immune system. This kind of disease is caused due to what is that? ADA deficiency or SCID. caused due to the effect of deletion of ADA gene. For the production of this kind of enzyme, ADA enzyme, ADA gene is very necessary. Usually, that ADA gene present particularly in the case of lymphocytes. The lymphocytes which produces adenosine deaminase enzyme. That adenosine deaminase enzyme is necessary for normal functioning of immune system. Here, this kind of disease, ADA deficiency or SCAD disease, is caused due to the deletion of ADA gene. For the treatment of this kind of disease, ADA deficiency, from a patient, the lymphocytes which are extracted, to rectify this kind of disease, or for the treatment of ADA deficiency, the treatment of SCAD disease, from the patient, lymphocytes which are extracted. Such lymphocytes which are grown on, suppose these are the lymphocytes, which are grown on a growth medium. The lymphocytes which are grown on a growth medium. Outside of the body. And the gene is extracted. It means ADA gene or cDNA, complementary DNA for the production of adenosine deaminase. cDNA or ADA gene inserted into the cells. The ADA gene is inserted into the lymphocytes. ADA gene or cDNA inserted into the lymphocytes with the help of one of the vector is used, that is called as retroviral vector is used for the transfer of ADA gene into the lymphocytes. Then these lymphocytes, which are modified into genetically modified cells or genetically engineered cells or transformed cells. Such transformed cells which are again re-injected, genetically modified lymphocytes again re-injected into the patient. Here in a patient, the ADA gene is entered, then there is a production of ADA enzyme. Then ADA deficiency is rectified. SCAD disease can be rectified. Here the transfer of genetically modified lymphocytes to the patient. In a patient, ADA deficiency can be cured. ADA deficiency or SCAD can be cured. But here, genetically modified lymphocytes not immortal. Here, not immortal means lifespan is very short. Genetically modified, usually lymphocytes lifespan is very short. Therefore, patient required periodic infusion. It means regular treatment is necessary to avoid SCAD disease or ADA deficiency. For the permanent cure, for the process of permanent cure, the case of gene therapy, the ADA gene extracted from bone marrow cells. The ADA gene extracted from the bone marrow cells, and again such genes which are introduced into the lymphocytes stages, early embryonic stages, the genes which are inserted into early embryonic particularly in the case of lymphocyte, it is a process that is permanently growth. Here this is the repeated treatment, particularly in the case of gene therapy. Here the gene therapy means it is a technique in which replacement of defective genes or fault genes by a normal functional genes to correct, to rectify genetic diseases. That is the term that is called as gene therapy. Here the gene therapy, the first gene therapy given in 1990 to the 4-year-old girl. The 4-year-old girl suffered from ADA deficiency. If the person shows ADA deficiency, causes a syndrome called SCID. Here the ADA enzyme is, it means adenosine deaminase enzyme is very necessary for normal functioning of immune system. This kind of enzyme is necessary for functioning of lymphocytes. Hence, from the patient, lymphocytes were extracted from the blood or a bone marrow. The lymphocytes were extracted from the patient blood or bone marrow. Such cells, lymphocytes, which are grown on a growth medium, outside of the body in a laboratory condition, then ADA gene or a foreign gene, or in other words, complementary DNA is inserted into the lymphocytes by using one of the virus. That virus is called as retrovirus. Retrovirus acts as a vector. Retroviral vector is used for insertion of or transfer of ADA gene into the lymphocytes. Afterwards, the lymphocytes becomes transformed cells, genetically modified cells, or genetically engineered cells. Such cells which are re-injected to the patient. Here the ADA gene produced in the patient. ADA gene produces adenosine deaminase. Then SCID is rectified. Adenosine deaminase enzyme is produced. The deficiency is avoided. Here this kind of disease can be rectified or cured. Usually, genetically modified lymphocytes, which does not show not immortal. Therefore, the patient required repeated treatment, repeated infusion is necessary for avoiding of that disorder. One more application in biotechnology application in medicine. One more application, that is molecular diagnosis. The molecular diagnosis is the method in which early detection of pathogens. It is the technique. Molecular diagnosis is the technique. In this technique, early detection of diseases which are necessary for early treatment. For the early treatment, early detection is very necessary at molecular level. Early detection is very necessary for early treatment. Particularly in earlier days, detection is not possible. The disease detection is not possible in conventional method. In early days, detection of diseases not possible in conventional method. Therefore, biotechnology, by using biotechnology, some of the techniques which are used for early detection. Usually early detection is not possible in the case of conventional method. Therefore, by using biotechnological process, early detection, the techniques which are used for early detection of different human diseases. Here, by using some of the methods, occurrence of early detections includes PCR technique, rDNA technology, by using rDNA technology, and by using the ELISA test. Particularly in the case of polymerase chain reaction, presence of virus or bacteria, presence of virus and bacteria pathogens, presence of virus or bacteria pathogens in low concentrations at the time, in this time are not visible the symptoms. The symptoms will not be visible in low the pathogens which are present in the blood, in low concentrations. The symptoms will not visible, and by using this technique, this can be, the disease can be, pathogens can be detected by amplification of nucleic acid by PCR method. By using this technology, the early detection of pathogens. And one more technique that is called as rDNA technology. In this method, muted genes in suspected cancer patients. Muted genes in suspected cancer patients can be detected by hybridizing test probe or a probe with complementary DNA in the cells of the patient, followed by autoradiography. By using this method, the cancer causing genes can be detected. The muted genes in the suspected cancer patients can be detected by hybridizing the test probe or radioactive probe, hybridizing the probe with complementary DNA in cells of the patient, followed by autoradiography. By using this technique, early detection of cancer causing genes. And one more technique for the early detection of diseases, ELISA, enzyme linked immunosorbent assay. By using this technique, detection of antigen or antibody synthesized by the body against specific antigen. By using this technique, the antibodies which are used to identify the specific antigen. In this method also there is early detection of pathogens, takes place. Therefore, ELISA test. These are the methods which are used in molecular diagnosis. The molecular diagnosis, these are the techniques in which early detection of pathogens, early detection of viruses, early detection of bacteria, early detection of muted genes, early detection of antigens can be detected by using methods, polymerase chain reaction method, recombinant DNA technological method, enzyme linked immunosorbent assay method. These are the techniques which are used for detection of diseases.